Welcome back, guys. Today we are going to be talking about active transport. Remember yesterday we talked about passive transport, which didn't require energy because things were diffusing naturally from high concentration to low concentration. So sometimes a cell is going to need to go in the opposite direction and kind of save up a whole bunch of one particular atom or molecule. And so we're going to learn about how that works and what that might entail. Okay, so active transport requires energy because we're moving against the concentration gradient. So we're going from low to high. So there are 4 ways that we usually do active transport. That can be a pump, it can be co-transport, exocytosis, or endocytosis. And we're going to get to all of those in depth later on in the video. Okay, so we know it requires energy, so that brings us to this molecule called ATP. So ATP stands for adenosine triphosphate. So if we look at this molecule here, you probably saw this in your macromolecule sorting activity. If we remember our functional groups, we know that the phosphates are over on the right-hand side. We have 3 of these phosphoruses with 4 oxygens around them. Okay, so figure out what's going on there. But if you look at the left side, that looks suspiciously like a nucleic acid. And we've got those double rings with the nitrogen in them. It's because it uses the same nitrogen base that some of the nucleotides do. So adenine looks like the word adenosine for a reason. And we have tri because there are 3 phosphates here. So this is our universal energy source for cells. Cells don't burn sugar directly. They use that sugar to make ATP, and then I use that ATP for all my cellular processes. And your body can turn a whole lot of things into ATP. So sugar, obviously, but also fats, and it can do it with proteins to an extent as well. So when people go on like the keto diet, they're actually using fat to make ATP instead of sugar. And that's why you can't eat any sugar, because your body wants to do that more than anything. Anyway, so we use this ATP for active transport. What it can do is the terminal phosphate group, terminal means the last one, the end of the, we can take off that last phosphate group, and it says it goes to the transport protein. So it attaches to it, which changes the shape of the protein to move a substance. So that energy in that phosphate group, it can alter the shape so that it can do something differently. So remember that energy is often stored in atomic bonds. So what we're seeing here, when I break off that phosphate group, it releases energy, and that's generally how we do with ATP. And if I break off that phosphate, it only has 2, so what would I name that? It's adenosine diphosphate, because remember, di means 2. So remember that when it's changing shape, like we learned last time, that's a conformational change. Okay, so let's talk about pumps. So sometimes I want to maintain something called membrane potential, which basically means that I am not wanting equilibrium. I want to have a difference between the inside of the cell and the outside of the cell, because this can make an electrical charge. There are a lot of your important body systems that rely on this. So your nervous system and your muscular system definitely use these pumps. So we're going to pause this PowerPoint for a second. I'm going to tell you a story. So I once went to a roller derby tournament with my friend in Baton Rouge because she's big into roller derby. And if you don't know what that is, it's a bunch of people aggressively roller skating, and sometimes they like bump into each other, like, you know, elbow and all that. It's pretty wild. Anyhow, so one of these girls she collapsed screaming, and to explain why, you have to understand what was happening. So when you exercise a lot, you lose salt through your sweat, right? But you also need that sodium to make your muscles and your nerves work. So this girl lost too much sodium and it made all her muscles clench up and she was in excruciating pain. This is why when you exercise, you have to drink like Gatorade or something. Or if you're really bad off like she was, you need salt tablets. And that's what they had to do to finally fix it. So this sodium stuff, it's no joke. You actually need some of it to live. Anyway, back to this. So the cytoplasm is relatively negative compared to extracellular fluid. So if I want a charge, I have to work at it. So some examples of pumps: electrogenic pumps generate voltage across the cell membrane, which can be used as an energy source for other cellular processes. So breaking that word down, electro is like electricity, right? And gen means to create. So I'm creating a little bit of electricity, which is kind of cool. So the sodium potassium pump, this one is super important because 3 sodium get pumped out, 2 potassium get pumped in. But this is how muscles and nerves and all of that work. This is the electricity they measure in an MRI in your brain, comes from this. All right, another example is a proton pump used by plants, fungi, and bacteria. We're gonna see this when we get into cell energetics. This is more like what we see in photosynthesis and that kind of thing. Okay, so how a pump works. Animal cells have a relatively high concentration of potassium inside the cell compared to outside, just naturally. Okay, so we have a relatively low concentration of sodium. So in this shape, the protein has an affinity toward sodium. So sodium is going to tend to attach to this pump. So ATP phosphorylates or detaches one of those phosphate groups to the protein, which causes a shape change. And so when the shape changes, I open up the enzyme to the outside environment. It's going to release the sodium and then potassium can get inside. So when the potassium binds to the protein, the phosphate group gets released and then it changes shape. And that's how this pump works. So if it's like down this way, okay, then we can have sodium will attach to it. But when I change shape and go like this, the potassium attaches to it when it's in that shape. Because remember, shape equals function. If I change the shape, I'm changing what it's doing. Okay, so co-transport. Like co-parenting means we're doing it together. So with co-transport, I am diffusing a molecule attached to something else. So something might have favorable movement or it's naturally diffusing downhill. So something's trying to diffuse, so I'm gonna piggyback something else to that thing that's naturally diffusing. So plants use co-transports for sugars and amino acids. So there is a sucrose hydrogen co-transporter. So sucrose can only travel against its concentration gradient if it's coupled with hydrogen that is diffusing down its gradient. So this could be important if we think about it. Think about what plants do with their excess sugar, right? They tend to store it in a tissue. So this would be like root vegetables, like a potato, right? All that starch is a whole bunch of sugar it bound together. Well, how am I going to get all that sugar down there, like enough to build it up? So I have to be able to go against the concentration gradient. Otherwise, all that sugar wouldn't stay in the roots and try to go to the rest of the plant. So that's how potatoes get all that good energy in them to make them delicious. So here is a drawing to kind of help us figure it out. And sucrose says, I can't go through there alone. And hydrogen says, I'll help you. So all these hydrogens, they're moving through maybe a hydrogen pump. Well, these sucrose molecules can piggyback and come with them. Okay, so if I want to transport something big or a lot of something at one time, sometimes I can't mess around with pumps or hand-holding diffusion or any of that business. So we go to exocytosis and endocytosis. So exocytosis, EXO, means out. Like if I exit, I'm going out. So I am sending something or secreting it through vesicles. So this might be, let's say there's, a gunshot or a clap of lightning, okay? You hear that response and then your adrenaline starts pumping. Well, there is a cell that has the instructions to make adrenaline. And so it'll make the adrenaline, it can't wait for it to diffuse. I gotta run away now. So it's going to dump it out of the body through exocytosis. It'll make a bunch and just dump it out. We're not even messing with diffusion, just dump it out. So these vesicles fuse to the membrane, and then when they do that, they release whatever's in there into the outside environment. So they use neurotransmitters like adrenaline, or say dopamine, like you get a big hug or you see a really cute dog. That rush of good feelings, that's from your nerve cells just dumping those good feeling hormones into your body. Okay, so endocytosis is when I take things in, like enter, endocytosis. So it's the opposite of exocytosis. Things can be absorbed wholesale. So we have phagocytosis or pinocytosis. And y'all are gonna laugh, but this is how I remember the difference between phago and pino, okay? I think of fat kid cytosis for phagocytosis. This is cell eating. A phage is something that eats, okay? There's, I'm trying to think of the word, some words that go with it. Anyway, but it really, fat kid cytosis. And pinocytosis is like pina colada cytosis. It's cell drinking. And then there's also something called receptor mediated endocytosis. This is going to be like what your immune system does. So phagocytosis, we engulf larger particles to be later digested by lysosomes. So remember that lysosomes have all those digestive juices in there. It can break apart organelles, but also big particles of food. So the cell surrounds the particle with pseudopodia. So if you look at the diagram, what that basically means is it can Pac-Man some stuff. It just it engulfs it, and then it makes a little vesicle. Like it pinches off some of the cell membrane to make a little pouch, and then I can attach lysosomes to it later to break it up. So the food vacuole gets fused with a lysosome to be digested. So pinocytosis is nonspecific uptake of extracellular fluid containing dissolved molecules. So if I notice that there's a whole bunch of, I don't know, dissolved salt or sugar or something I want, I can surround it just like I did with phagocytosis. Sometimes they'll use a protein-coated vesicle to do this so I can transport it better. Okay, so phago is eating, like a food particle. Pino, we're uptaking something from the fluid. Okay, so receptor-mediated endocytosis. So specific uptake of molecules via solute binding to receptors in the plasma membrane. So something can trigger endocytosis if it's the right stuff. So it allows the cell to take up large quantities of a specific substance. So if I specifically need sugar or specifically need salt, I may use this route instead of just regular pinocytosis. So this is kind of like a more specific version of that. But also, this is kind of how your immune system works. I think this is, yeah. So let's talk about this for a second. This is how they're called phagocytes. No, macrophages, there we go. They're called macrophages. Macro means big, phage means eating. So you have these big white blood cells that swim around your bloodstream. I mean, they're relatively big. It's not like you're gonna open your vein and like a big old honking one's gonna pop out. Anyway, so if they find something that's been marked for destruction, like a bacteria, it will attach to that receptor and it will eat the bacteria. And it digests it just the same as you would any other food particle, which is kind of cool. Don't get me talking about microbiology, y'all. I could talk about it all day. Okay, so I'm pretty sure that is all we've got today. For your assignment today, you should be using the rest of your science time to work on your cell campaign materials. The next thing that's due is your smear campaign pamphlets. Remember, each person in the group has to make one. Like, I need one for every person there. So don't just foist that all off on one person. Everybody can make one, and everyone has to make one. And everyone should make one on a different candidate. So if you're a chloroplast, I don't want you all to do the nucleus. Everybody, like, one person does nucleus, one person does ribosome, one person does mitochondria, whatever. Those are going to be due at the end of the day tomorrow. So you can work on it today in class, and you can work on it Friday. Remember, that can be digital or print. I just make sure it gets turned in, and that you're not leaving this all to the last minute. This is a 3-stage project. You need to work on it. So just let me know if you have any questions about what is expected or about this content, and I'll see you guys in the next video. Take care.